My name is Joseph Wiemels, and I'm a professor at the Center for Genetic Epidemiology at the University of Southern California. My research investigates the causes of childhood cancers. In this presentation, we will focus on the genetics of childhood cancer, how scientists identify genes that influence cancer risk, and how genetics can help define those of us who are more vulnerable to environmental risk factors. When we refer to germline genetics here, we are including those genetic variants that we inherit from our parents, and also rare mutations that may occur in parental germ cells or in the zygote soon after fertilization. How do we know when diseases have a genetic cause? Scientists use family studies to calculate heritability of diseases the same way that we can evaluate genetics behind other common traits, like hair color or nearsightedness. Many common conditions appear to have a strong genetic influence, noted by the high risk of disease among siblings of individuals diagnosed with, for instance, autism or diabetes. We do see increased risk of cancer in siblings of children with cancer, but comparatively with these other conditions, this sibling risk is far lower, here shown to be only 2 to 3 times greater for siblings of children with cancer compared to the background risk in the general population. Hence, there must be other important causes not captured in genetic factors, namely environmental factors. There are 2 types of studies that examine genetic factors and the risk of cancer. The first of these are family studies, shown here on the left, where we find multiple members of a nuclear family that have the same or a similar pattern of cancers. This usually indicates the presence of a strong germline predisposition variant, one that was passed down to family members from parents to children. These types of strong mutations are rare, and so are the cancers that result from them. Most cancer is sporadic in the population, and the genetic influence is detected via case control studies shown here on the right. In these studies, participants are unrelated, and we are simply looking at the association of common polymorphisms that differ in frequency between normal healthy people and those with disease. Let's talk a bit more about these 2 study designs in the next slides. Because of our ethnically diverse population here in California, we are able to compare familial risk by comparing clustering of childhood cancer within families between different groups, here described as non-Latino whites, Latinos, non-Latino Asian Pacific Islanders, and non-Latino blacks. We found that U.S. minority groups had higher risk of cancer in siblings of a childhood cancer case when compared to non-Latino whites, and this was significantly so for hematologic cancers in Asian Pacific Islanders and solid tumors in blacks. Note that not all this risk can be assigned to genetics, as family members are also living in the same house, so they share environmental factors as well. Familial risk is most often caused by very strong singular genetic mutations. These are called genetic predisposition mutations, many of which are associated with syndromic conditions. Such cancer-causing mutations are either inherited from the parents or result from mutational errors originating in the child's germ cells. In these cases, the cancer can be attributed almost entirely to the presence of the mutation. We have found such mutations in about 40% of the children from families that have multiple members with cancer. These families represent, however, a small proportion of all childhood cancer cases, most others likely having a stronger environmental component to their risk. Genetic susceptibility also plays a collaborative role in childhood cancer more broadly and in context with environmental factors. Here we show a series of common genetic variants that are not causal by themselves, but weakly contribute to leukemia and are discoverable by comparing large numbers of children with cancer with healthy controls analyzed in a genome-wide association study. Interestingly, when we add an environmental exposure to this analysis, in this case tobacco exposure during pregnancy, a host of new genetic factors reveal themselves, indicating that there are critical genetic vulnerabilities within some children when exposed to toxic environmental exposures. This is an example of a gene-environment interaction. When we look at the multitude of exposures linked to childhood cancer, we can see that these are quite common. So why is cancer so rare? It is rare because genetic vulnerabilities exist in some children, while strengths exist in others that may protect them from the same exposures. In addition to this, pathways to cancer are long, multifaceted, and have many checkpoints. When these checkpoints fail, we end up with cancer, and oftentimes we can link specific mutations in the cancer cell back to the exposure in the same way a forensic scientist can explore a bullet wound. This pathway to cancer is complex, and delineating the pathway requires collaboration between exposure scientists and geneticists to discover the causal pathways with the hope to prevent cancer in children in the future. We'd like to wrap up this presentation with a few key concepts. First of all, germline genetic variation, those are variants that we've inherited from parents or mutations that have occurred in germ cells, can influence the risk of cancer in children. Strong or high penetrance germline alleles are responsible for cancers that run in the family. Common or low penetrance genetic alleles can impact our children's risk of cancer and contribute to vulnerabilities to environmental exposures that we know of as carcinogens. So, genetics are clearly important in cancer risk and are a major reason to explain why some children contract cancer and others do not, even when exposed to the same environmental risks.